Welcome everybody, all the attendees to this Discover Brigham session, what we know about COVID-19. My name is Gerald Peer. I am the moderator for this session. I am a microbiologist at the Brigham, as well as a professor of medicine, microbiology, and immunology at Harvard Medical School. I have been here for over 4 decades. My research focus has been on vaccines for a number of different microbial pathogens, in addition to other aspects of how we respond to infections and try to control them. We hope to deliver to you today a lot of updated information about the current status of the COVID-19 pandemic, which is clearly dominating the news. I'm joined today by 2 excellent co-presenters, Ann Woolley and Athy Sibris, who will also be discussing various aspects of the Brigham's involvement in the response to COVID-19. So let me ask Ann to introduce herself and tell you a little bit about her background and area of interest.
Thank you, Jerry. So I'm Ann Woolley. I'm an infectious disease physician at Brigham Women's Hospital and Dana-Farber. My focus within infectious diseases is on the immunocompromised patients, those that have undergone a transplant or oncologic treatments or bone marrow transplants. And I'm also a clinical investigator and clinical trialist, and then have been working the past 9 months, as all of us in our division and in many other divisions as well, focusing on some trials with COVID-19.
And Athy?
Yeah, great. Thanks, Jerry. My name is Athy Sibris. I'm one of the infectious disease doctors and virologists here at the Brigham. Before COVID, my lab focused mainly on the study of HIV. And like many people around the Brigham, we've been asked to contribute mainly clinically for me, but a little bit from my lab towards helping to fight the COVID pandemic here in Boston and around the world. If I may, Jerry, I'll start sharing my screen and then we'll begin today's presentation. Let me just say on behalf of all of us, good afternoon and welcome to today's session. I'm going to start us off talking really here about virus-host interactions during COVID-19. And just to make sure we're all on the same page, there has been a lot of discussion over the summer about when the second wave is coming. Is this the second wave? No, that was the second wave. Please, we can end the speculation. The second wave is happening right now. This is a screenshot taken from the New York Times looking at cases currently, and you can see that we are now approaching levels of cases that are very similar to the surge we had back in the spring. So this is a very good time to review what it is that we have learned since the spring, what lessons are there that we can glean from patients who have had both severe COVID, but also mild COVID disease, and how will that help us take care of future patients. So specifically here, I'll start talking a little bit about how COVID and SARS-CoV-2 came to the United States. What have we learned? Has the virus changed during the pandemic? Yes or no? And if it has, how has it changed? And then we'll ask some questions about when people have severe disease, why do they have severe disease? And then look at the other side of the coin and end on a more positive note. For people with mild disease, what is it that gives them milder disease? So let me then switch and start us off by taking us back to the beginning of what has been a truly glorious year of 2020. As we are all aware of, COVID came from Hubei province in Wuhan. And at least in terms of how it spread to the West Coast, the best available research, this is now phylogenetic and molecular clock analyses, suggest that SARS was introduced and propagated on the West Coast into the Seattle area around the end of January or the beginning of February. This portion of the slide also makes the point that there have been several introductions into the United States earlier in January in the Seattle area, later in February into the San Francisco Bay Area, that did not significantly propagate and contribute to the pandemic, making the point that it is possible with contact tracing, with aggressive efforts to limit the spread. In terms of what has happened on the East Coast, as you also know, COVID came from Wuhan to Italy, spread throughout Europe, and then came to the United States. Boston did see an influx of cases mainly from Europe, but in fact also from New York, as you're well aware. And researchers now estimate that there have been perhaps 80 separate introductions of COVID into the Boston area. And this is work done by Jake Lemieux, a former Brigham ID fellow now at the Broad Institute, and really makes the general point here, which is what I want to emphasize, that intensive public health measures that you put in place to surveil before the pandemic are much more effective than those efforts that are put in place after. Recognize here that there are limitations. So as an example, if you look at Great Britain, they estimate that COVID has entered the country 1,000 separate times. And so there really is no surveillance measure alone that can protect you against an onslaught of that magnitude. In terms of how we think about SARS-CoV-2 in terms of is it changing or not, I just want to take you through and get some measure of the diversity of these different viruses. And we're going to start off here with an example of HIV. And as you can see here on the left side, we measure the diversity in what are called these phylogenetic analyses. And you'll see here many branches, many leaves off the branches. And the more distance there is here, the more diverse the virus is. This is important because if you try to make a vaccine to say these viruses here at the top, there's really no guarantee that they'll work against these viruses here at the bottom. These are all HIV strains across the world. If we look at an individual patient, there is less diversity. And similarly so for flu. So remember here, flu is a virus we can make a vaccine to, but we have to change it from year to year because of drift and shift. This is the amount of diversity present in SARS-CoV-2. And some of you right now may be wishing you had better resolution on your monitor, so let me blow this up 10 times. And you'll see that the amount of diversity here remains actually very small. The point I want to make here and highlight for all of you is that this virus is a sitting duck for vaccine development. So while we should not take press releases from pharmaceutical companies at face value, we should evaluate the data ourselves independently. This really is a very good target for vaccine development, and we do take some hope from this virology. Now, this does not to say that the virus has not changed at all. What you may have heard over the summer, if you were looking in the lay press, is that the virus had mutated. There was a super virus. And they talked a lot about this mutation D614G. So what does that mean, and is that important? Is there a super virus? What I'm showing you over here on the left is the virus machinery. These are virus proteins that it uses to enter cells. And at the bottom here, you can see that the virus entry machinery, this is the spike protein. Spike proteins interact with the ACE2 receptor on cells, as you may have heard. Your immune system absolutely despises these proteins, and so the virus knows this and tries to coat itself in a shield of sugar, a sugar shield, if you will, to sort of avoid the development of antibodies. This D614G mutation here is what has changed, and as you can see here on the right-hand slide, really once the G virus showed up, it entirely took over. So should we be concerned about that? Research here in the last couple of months has shown us that this G virus is in fact more fit, meaning it is better at its job than the D virus. It increases the stability of the SARS-CoV-2. What that means is these spike proteins, actually, I've shown them here and they look very well structured. This whole apparatus really is inclined to just fall apart. And so when I say the virus is more stable, the G virus, this protein is less likely to fall apart. It can produce more virus in the upper airway, so that sounds concerning. But what we've also noticed, it does not appear that this G virus has more morbidity or mortality. It still gets neutralized by the exact same antibodies, and so it is less likely that the G614 mutation will reduce vaccine effectiveness. And in fact, all the vaccine strains were made with this D virus, and at least with the Pfizer data, they seem to have worked fairly well. Now, what are we going to learn here from patients who have had severe disease? On the left-hand side, we're just going to take a little bit of a detour here and point out the fact that there are archaic hominins, more ancient humans, that have contributed to our chromosomes. Specifically here, you have Denisovans and Neanderthals, and there has been genetic admixture, at least with Asians and Europeans, such that about 1 to 4% of our genome comes from Neanderthals, comes from Denisovans. When it came to COVID, people tried to do these genome-wide association studies, a GWAS study, and asked the question, are there genetic contributions to severe disease? And in this case, we defined a severe disease as disease with respiratory failure. So the patient needs oxygen, the patient has to go on a breathing machine. And they find that there is an association with some genes, some alleles on the 3rd chromosome. When they go and look here on the right-hand panel at zoom in on this region of chromosome 3, they notice that the most important area is a region that is entirely derived from Neanderthal DNA. This region is present in about 4 to 8% of Latin Americans and Europeans, but it's a third of Bangladeshis, and most importantly, 0% for African and East Asians, and we know those populations are certainly susceptible to COVID. So this does not explain the entire disease, but we will make the point here, the major genetic risk factor for severe COVID is inherited from Neanderthals, not from Denisovans. And the function in this area, what do these particular genes do? It's not entirely clear. Maybe they protected against ancient pathogens, but more work will be done on that in the coming months. What about the flip side of the coin? As I mentioned before, what can we learn from mild disease? This is a study that was actually conducted at the Brigham and published very recently from Dwayne Wiesman's lab. And they looked at these quick healers. They looked at about 90 participants who were mostly women and who were nearly all white. And they asked the question, how do these patients get better when they get better? I would just make a point here that it is no fault of the researchers that their samples mainly came from Caucasians. They used the samples they have available. But I think this is an important area in terms of clinical research equity. And so I would commend to you Dr. Busola Ojikutu's talk later this afternoon. She has been a leader at Harvard and really across the Boston areas, specifically as it relates to Black communities and engagement in research. So back to the study here. What I'm showing you here on the left panel is that in these people who all got better, almost all of them did not need to be admitted to a hospital. You can look at the different virus proteins, and in all cases, the patients make antibodies. In some cases, they make a little bit of antibody. In some cases, they make a lot of antibody. And it's not clear how much antibody you actually need to be protected. What they did look, though, if they looked at these patients over 5 to 6 months, they asked, in people who maintain these levels of antibodies, what type of symptom duration do they have? And if you could maintain your antibodies, you actually had shorter durations of disease. And so this gives us a hint as to what protection antibodies may be giving us. In the lower left-hand panel, I'm just going to make the point, without going into a big immunology lecture, that you have B cells that make antibodies. You have T cells that will help B cells, but they will also attack viruses. And the point here is that even sometimes when the immune system, the B cells forget and they lose the antibodies, the T cells remember. And this is seen in people after they have COVID. This is seen in people with asymptomatic disease. And so the point here is that T cells are augmenting the immune system. And so really some questions here that we're left with from this work is, what really makes a sustainer? And we don't really know that. Is it that they perhaps were exposed to other coronaviruses, which have offered protection? Or is there some other features of their immune system that are important? And then the question here, which really will be answered in a lot of the vaccine studies, is how much antibody response is enough? How much T cell response is enough? So what I hope I've given you here very briefly is just an overview of virus-host interactions. I think it's important that we review and summarize really what are the latest cutting-edge updates to treatments, to testing. And for that, I'm going to stop sharing my screen and I will turn things back over to Dr. Ann Woolley.
All right, well, thank you very much for that. I'm now going to transition us into thinking on the more of the clinical realm as far as what do we do with how this, now that we are, as Avi referred to, not wondering when the second wave is coming, but the fact that we are in that. How are we managing that? So I want to focus the first few minutes of my talk on testing and diagnostics, and then want to focus the second half on the therapeutics and all the developments we've had over the past 9 months with treatment for COVID-19. So first, just to start with saying, how do we think about testing? Who do we test and what? We all know that the variety of symptoms that anyone can have, and it's not necessarily the fact that one person having certain symptoms such as upper respiratory symptoms compared to those that might have gastrointestinal symptoms are going to have necessarily a different course. But we do know it just makes it even more challenging to understand, particularly as we get to the winter months, which symptoms should alert us to this. And we know that the mean incubation period is 5 days prior to those who are going to have symptoms for them to display it. We also know and have just learned significantly in the past few months just how prevalent asymptomatic disease is. And not only is the fact that so many we estimate and have modeled are asymptomatic, but it's also those that are actually causing transmission are thought to be asymptomatic at that time, anywhere of around 40 to 45% of the cases of transmission. And we're learning more and more now as to what the longer term sequelae of COVID-19 is, 30 days, 3 months, and now 6-month data that we're coming to understand. But what this doesn't help us to know is, well, then who should we test? If we don't have the capacity to test everyone, and fortunately we're in a much better situation than we were in March in this country, however, we still have significant issues with testing and bottlenecks. So who do we test and how do we decide who is the best ones to test if we do have some limited constraints on testing? So if we think about the disease course here, from the incubation period time to those that then have symptomatic disease, and then afterwards as they are then convalescing, we here need to know that we want to be sure that we are focusing on really parts because we want to be able to diagnose people earlier, do contact tracing, and then try to prevent transmission and therefore keep people away from actually getting to that symptomatic and more significant course in their illness. So what are the testing that we have for it? So very simply, we have molecular tests, PCR, LAMP, CRISPR, antigen tests or the rapid tests, and then antibody tests. I'm just going to very briefly touch on antibody tests to say, at the end of the day, we know a lot more now than we did in March as to what it means to have an antibody, but we still don't know the answer. All we can say is that by having an antibody, it means you've been exposed. It does not necessarily say that you are immune, and if you are, for how long that will last. But let's focus on molecular tests and the antigen tests. So we know there's a number of rapid antigen tests that have now received emergency use authorization from the FDA. This is a picture of the Binax Now that was approved at the end of August. There's a lot of benefits to this, and we know that there are a lot in our public health field that are really trying to advocate for doing more and more rapid testing. It's inexpensive, it's point of care, a short turnaround time so we don't have these bottlenecks of waiting days to get a test result. But there's also a lot of limitations. And there's been such a focus on the fact that they have a lower sensitivity compared to the gold standard, the PCR test. But what we really need to take a step back and see, who do we want to diagnose? Do we want to diagnose anyone that just may have some SARS-CoV-2 RNA? Or is it really that we want to diagnose those that have enough virus that is infective and then could actually transmit virus? And if that's the case, should we not be okay with having a lower sensitivity with our testing? But there is a flip side to this. Given the fact that overall the prevalence is still in the lower end of things, we can't forget the fact that there are also going to be false positives. And what does this mean? If we employ this then, let's say in school testing, how will that only cause more hysteria and also bottlenecks, because then to have to confirm these tests with PCRs. So there's still a lot of unknown, and currently these that are approved for rapid testing do not have approval for asymptomatic screening tests yet. So when we think about molecular testing, in the middle we have the gold standard that I'm sure many of you have had to endure, which is the uncomfortable nasopharyngeal swab. And then on the left we have the anterior nasal swab, which fortunately many places around the country, ourselves included, are going to, which is a much more comfortable way of being able to obtain a sample. And then to the right, saliva, which is now being used more and more, though there's a lot of difficulties on the lab side of thing from being able to actually process these samples and do it in a high throughput way. But the question everyone has with this is, are these equal? Is it the same, or is nasopharyngeal the gold standard? What about a lower respiratory swab? Well, we know here, and I should depict here the study that shows about saliva compared to nasopharyngeal PCRs, and that they have been validated that they are as good as nasopharyngeal as have the anterior nasal swabs. I'd like to just highlight here a study that myself and two of my colleagues, Deb Hung and Lisa Kazemi, are running in order to really try to answer this question of how do we get through this bottleneck of testing? How can we get testing to individuals at their home? And so we're running a large study in collaboration with the Broad Institute to enroll 10,000 participants in the greater Boston area, and really trying to empower patients to be able to provide access to testing, particularly in these underserved communities that have been disproportionately impacted by COVID and have only had amplification of the already health inequities that had existed prior to March. We want to be able to see what is a seroprevalence of COVID-19 in the greater Boston area, and then use this as a way to develop a model to implement large-scale at-home testing, not only for COVID-19, but as we get into the winter months, for other respiratory viruses as well. We're doing this with paired testing, both an anterior nasal swab that is self-collected at home, as well as a dried blood spot, which we then test the antibody. So now I'd like to just transition at the end into treatments. So we know there have been many therapeutic classes that are under investigated, and we have hundreds of trials that are currently on clinicaltrials.gov, and many, over close to 200 that are awaiting some, or have put in applications for different EUA approval with the FDA. Let's think about it in categories. So first we have our antivirals. So there's many here, some that we have number of studies to show that they do not have any efficacy, but we fortunately early on learned of the efficacy of remdesivir. They've had many studies. The NIH placebo-controlled trial was the first one that came out this summer to actually show that it did decrease duration of symptoms and duration of stay, though it was not powered and did not show, based on statistically, that it had a mortality benefit. We have perhaps the biggest category of tests, or of treatments, excuse me, are immune modulators. And I'm highlighting here dexamethasone, because as we're all aware, that has really took many of us by surprise, the fact that this was the first treatment with COVID-19 that did have a significant mortality benefit. And I'll get to that in our next slide. And then what about neutralizing antibodies? Well, we've been hearing more and more about this over the past few months, and fortunately, we now have the one by Lilly that has received an EUA approval. Though there's still a lot of skeptics as to understanding who and when is it best to use this, and what really will be the benefit of this. What we do know now is that the earlier in disease that you use a monoclonal antibody, they're more likely to have an impact. I put a question mark by convalescent plasma because though that has received an EUA approval, there's still a lot of skepticism as to who should use this and does this actually have significant efficacy. So I just want to highlight two of the large trials, the recovery trial, the UK trial that has enrolled over 11,000 individuals from over 175 different hospital organizations in the UK. You see here in red the different treatments that has been tested, and it's done it in a very unique approach in being able to really ramp up clinical trials and enrollment despite it being a pandemic, and they did this really from the start. And it's from the recovery trial that we learned that dexamethasone did have a significant benefit and a mortality benefit such that they halted the arm of the dexamethasone arm in that study in June. The WHO Solidarity trial is an adaptive open label trial that's being conducted in over 30 countries and again over 11,000 participants. And you see here the different drugs that are being used, and here remdesivir is used as compared to not shown in the recovery trial. And unfortunately, we learned from the Solidarity trial not too long ago that there actually was not seen to have a benefit for remdesivir as we had seen in some of the other trials. And so there's a lot of confusing information, I would say, if you will, on remdesivir, that we know it helps, but who does it help and how much, and does it actually lead to a mortality benefit? So I'd like to just end with putting some of the key therapeutics in back to how we started this when we think about the progression of COVID-19 infection. So we have stage 1, mild or pre-symptomatic, the symptomatic, perhaps hospitalized, requiring some oxygen. And then that stage we all want to try to prevent patients from having to enter, which is the hyperinflammation phase, where ARDS and shock. And so when we think about our 3 main treatments right now that we know of, we think of remdesivir as that sweet spot in the middle. Someone's on some supplemental oxygen or minimal, but yet not sick enough to require mechanical ventilation and be intubated. Whereas dexamethasone, we know was not shown to have benefit early on in disease, but did have mortality benefit in those that were severely sick with COVID-19. And then fortunately now there's been much more attention brought to how do we actually prevent patients from getting into the hospital? How do we actually treat them as soon as we're able to diagnose them? Now the testing is so much more available that we can diagnose earlier and contact trace and then hopefully treat. And is monoclonal antibodies the answer? Well, an IV infusion is obviously not the key to what we would like to have for early treatment, but at least we are now seeing that we have something, and there's so many more studies that are currently still under trials. Thank you for this, and at this point I'd like to turn it over to Jerry to talk through vaccine development.
Okay, thank you very much. Okay, I gotta unmute myself. Can you hear me? Am I unmuted?
Yep.
Okay. And I am not able to move my screen. There we go. Okay, so I'm going to talk briefly about vaccines. We are running a little bit late, but there will be a networking session after this where questions can be answered if we don't get to them. So the good news is that in 2020, we have a number of approaches that we can use to make vaccines. You can see 8 of them listed here. I'm not going to go into detail. Some of these are tried and true, such as inactivated and live attenuated vaccines and protein vaccines. Others, such as the mRNA vaccine, which is the technology being used by Pfizer and Moderna and are out there early, are brand new and have a lot of potential, but very little experience. So where are we at today in terms of vaccination? Well, Russia has approved 2 vaccines, but not really based on any good safety or efficacy data. That seems to be a political decision. China has distributed a killed vaccine to the military and to some of their frontline workers, but again, little to no real data about safety or efficacy. And as of yesterday, I could find 179 total vaccines have been reported to be under development for SARS-CoV-2. 56 are in human trials, and 10 of these are in phase 3 trials. 2 to 4 of them, it's a little difficult to tell, have completed enrollment. And as everybody who has been paying attention to the news knows, on Monday, Pfizer BioNTech reported the results from their phase 3 study, although only in the lay press, and claimed greater than 90% efficacy of their mRNA vaccine. Okay, so a lot's going on, and a lot's going on here at the Brigham as well. I just wanted to highlight that there are 3 COVID-19 vaccine studies going on here that people within the community can participate. They encompass all 3 of the major approaches: the mRNA approach, the vectored adenovirus approach, and the recombinant protein approach. And it's very likely all 3 of these, if they work, will be part of the armamentarium we will have down the road. So what can we expect to get an emergency use authorization, which I want to emphasize is not a full license, from the FDA and deployment in the US? 2 companies, Pfizer and Moderna, the mRNA companies, expect to have enough data to submit an EUA application sometime in the next few weeks, okay? And what will we want to know? Well, we'll certainly want to know about safety, particularly the number of serious adverse events. We'll want to know about the immune responses, how good they were, and longevity, but that question on longevity will only take time to answer, obviously. And efficacy, how much less disease and what type of disease. Does it completely prevent disease? Does it lead to only mild or moderate disease? And according to what Pfizer reported earlier this week, we might expect a high level of efficacy to be the new bar. What's going to happen to be decided in order to start vaccination? So there are a lot of questions here that really get out of the realm of science and microbiology and immunology and into public policy. So there's the practical, the vaccine supply. As everybody's been reading, the mRNA vaccines have to be stored at -80 degrees centigrade. What about the supply? Pfizer's expecting to have enough vaccine in the year 2021 for only about 12.5 million people in the US, far short of what we'll need to have a high level of immunization to protect a large number of people. Stability, distribution, availability of personnel to administer the vaccine, availability of needles and syringes. Many practical issues will have to be solved. The difficult will be prioritization of who gets this. And again, this is being discussed openly. And as mentioned earlier, we really don't have a good answer. And the most challenging. This is going to involve not just the scientific and medical community, but political and public health individuals about safety, the need to vaccinate, and to get out the message as to the risks and value of vaccination. Okay. So what are going to be the major issues? Well, safety is going to be a major one, and how these impact and reduce the level of disease that occurs following vaccination. What will be missing in the early phases of vaccination, because we just won't have accumulated the data. We won't know about the long-term safety, immunogenicity, and efficacy until long-term occurs. We don't know whether development of protective memory responses will occur, such that even as antibodies wane, we're still immune. And we won't know the determinants of long-term protective immunity, again, until we've had enough time to study this. And then finally, while I could say all new vaccines are subject to this missing concerns, we usually have years worth of experience with vaccines, as you can see here, going back nearly a century, which support getting them used and distributed to the public because we have a lot of data, and much of this will not be available for a while. So, will the success of political and public health messaging be the major determinant here for the uptake of vaccines? Well, obviously, we need to get appropriate messaging, particularly to high-risk individuals and among members of traditionally underserved and marginalized communities. We need to be honest about the risks and benefits, and this is going to involve saying some things that maybe some people won't want to hear, particularly about the lack of data that we might have over long-term risks. We'll need to develop the tools for the public to use to assess their risks and evaluate their benefits to aid in the vaccination decisions, and this will have to be widely distributed. And we'll have to, of course, deal with the anti-vaxxers who are likely to be out in force. And we see that there's going to be a lot of components to successful immunization that will have to come into play from both the medical and scientific community comprising the immunization system, the healthcare workers who will have to distribute and deploy the vaccines, and decision makers that will impact the family and how families decide to accept vaccination. So how's it looking right now? Well, the warp speed that we all read about was truly that. It's likely to result in vaccines to be extensively deployed in the next 2 years. That's truly remarkable. Less than a year to get there. Early data suggests that vaccines may be better at inducing immune responses than occurs following natural infection, so that will be good if it holds up. Predictions that we might need about 60% of the community to be immune to achieve herd immunity are out there. This is kind of a soft prediction, if you will, but it's more or less a bit of a loose consensus, I would say. And then finally, if this does all come together, it would likely be one of the major achievements in science and medicine in the 21st century to deal with this pandemic in such a rapid and effective way. So I believe now we have perhaps a couple minutes for questions, and then we can go to the networking session. And I believe there should be a link to that networking session available to the attendees. So we only have about 1 minute left. Are there any questions? You put up in the chat here.
I think there's one that it's there.
Abby, a question for you.
Yeah, thank you for the question. Are the Neanderthal genes protective? So no, the Neanderthal, that area of the genome is bad. So that is the area that maps to the major genetic risk factor for severe COVID. So we don't know exactly what gene it is that's in there, but that region of Neanderthal DNA is a risk factor for severe disease. So that area is bad when it comes to COVID. It works out to be about half of South Asians, maybe about 15% of Europeans that have that particular allele, if you count people who are heterozygous and homozygous. So it's not going to explain the whole picture, but it is it's a bad risk factor.
Great, thanks. And there's one more question, Anne, for you, if you could look in the chat, and then we could go to the networking session.
Sure. It asks about if the mortality rate has decreased based on the improvement that we've seen in therapeutics and treatments, or is it just the fact that the demographics of who's getting infected has changed? I'd say the short answer is that we don't know for sure. I think we will need to wait to see. We know that when we are seeing increases in cases, we expect to see the increase in hospitalizations and mortality 2 to 4 weeks after that. And so I think the next 2 to 6 weeks will be very telling in that regard in this area. But overall, yes, I think what we've seen a big shift in is twofold. One, the fact that we're doing much more testing of asymptomatics, and so diagnosing earlier. And also we are having, therefore, the shift as far as the age and demographics. We're having younger people that we are diagnosing, and are therefore not all, but the majority less likely than those over 65 to go on to have more severe disease. But the hope is that treatments are having some impact, but I would not say yet that we have enough evidence to say that our therapeutics are making such a difference, even though the hope is that now from a hospital system perspective, in many places, but not all, we're not as resource constrained in the sense of being able to then be overwhelmed with that. And so I think it's a part of all of those. But again, I think we'll learn more in the next 1 to 2 months.
Great, thanks. Well, I'd like to thank my co-panelists for their excellent presentations. I'd like to thank everybody who attended and listened to this session. And now we can move on to the networking session via the link in the, I believe that might be in your browser.
Great. Thanks, everyone.
Thank you.